So the point of this is the hybrid that they, this is a chicken. At least that's how it is. So they get this hybrid out of this, so it looks like a mixture. Now it looks like a donkey chicken. Okay, so if you don't know Japanese quail, they're actually, probably most people are more familiar with California quail, which are bigger. Japanese quail are fairly small, especially the females. So you get this kind of hybrid as a product for the offspring in terms of the appearance. But the part that was important is they could use mitochondrial markers and show that in a hybrid, all the mitochondrial DNA looked exactly like Japanese quail mitochondrial DNA, not like chicken. So it was a way, by using actually a completely different species for the male and for the female parent, you can track where that DNA came from. So all the mitochondrial DNA in the offspring was identical to the quail in this case. And so they did a similar experiment in the horse-donkey mule hybrid thing. They would look at the mule, and if you knew the horse was the female mating parent and the donkey was the male, for example, or vice versa, you could then track the mitochondrial DNA in the offspring and show that it came directly from the mother, all the offspring. They could do this over and over and over again. Okay? And they actually use, just because you guys know what this stuff is, when I say it looked like, they could do this before sequencing because they actually use restriction enzyme digestion. Okay, so there's a way, and you guys don't need to be overly concerned with the process. But since you drew, or inaccurately drew, restriction enzyme maps on your first exam, right? Only I asked for a linear strand, but you guys have also thought about a bacterial circular strand. There are restriction enzyme sites. These are recognition sites, right? In the DNA sequence that this enzyme will cut there. That's almost like a barcoding pattern between species, okay? There are only certain types. These sites for these enzymes are found in different spots on each species. In fact, they're found in different spots between organisms, but they're fairly conserved within a species. So we can tell quail from chicken just by taking the DNA, cutting it up with a bunch of restriction enzymes and running it out in a gel and seeing different fragment patterns come out. Okay. So this was before sequencing or anything like that was available. So you can use a fairly simplistic molecular mechanism technique in the lab to show in all the progeny, specifically isolate their mitochondrial DNA, ignore the nuclear, and show it all came from the mother parent. And this was, of course, repeated over and over so that they prove maternal inheritance. So does that experiment make sense, at least on that level? Okay. And you guys also encountered in your homework this concept of a maternal effect. Okay, so this is where, because of maternal inheritance patterns in the mitochondria, your offspring, so the phenotype of the offspring, is dependent on the maternal genotype. So that's what we mean by maternal effect. I know my hybrid here looks like some kind of weird thing going on. So the main key thing here is the inheritance pattern for most organisms for mitochondria comes straight from the mother, and it's also an exact copy. So when I say looked like the quail mother, it's a perfect exact copy. Whereas your nuclear genome as offspring, that's a mixture, right? It's not an exact copy of either one of your parents. That's a great, you and your siblings are actually more alike genetically than you are to either one of your parents, okay? Because if you compare yourself, your genes, your DNA to your parent, you just have half, theoretically, of their composition of alleles and genes, right? Whereas you and a sibling, you each have half and half from one of your parents. So nuclear is very different than mitochondria. Mitochondria is going to be the exact same copy. There's no, barring any kind of strange mutations and things like that, mitochondria inheritance patterns comes exactly from mom and exact perfect copy into the offspring. No recombination or changes for the most part. All right. Why would this make sense? So why are most organisms, why do we see maternal inheritance? What's going on at fertilization and zygote? Yeah, so if you, where's my 242 students from last year? What's in the egg and what's in the sperm, right? We drew both of these things out. We had all kinds of labeling. You guys know what this stuff is. Jason's laughing. So we went into detail. You took 242. If you had any biology course, okay, let's just start basic. Size-wise, egg to sperm. It's way bigger. All the organelles are here. You have cytoplasm in this cell type. It's big, okay? The sperm is small. Really all it has is a nucleus and a few mitochondria for motility. Right, those are attached usually to a flagellum in animal species. So egg, large, has a nucleus, has a bunch of mitochondria. In plants, it has the chloroplasts. Okay. Has a whole bunch of other stuff going on. You have yolk, you have, and then as Carl said, all the other organelles. So when these two cell types come together, when you have a sperm and egg fuse at fertilization, the contributions are much greater from the egg towards the zygote in terms of composition. Okay? So but what about the mitochondria and the flagellum? How come those don't get contributed in?
They just fall away. They stay in a flagellum.
They just float away. Okay, so at fertilization, I'm still trying to find my little 242 students in the crowd. You're not gonna. Jason, what happens to the flagellum? You remember? There's somewhere with you, but.
I wouldn't have a correct answer.
Yeah, and
It's degraded.
yes. So most species, one of the ways to prevent multiple polyspermy, to prevent multiple sperm from fertilizing, the egg has a whole bunch of enzymes put in place that degrade the rest of the sperm, and especially the additional sperm that are in the environment, but especially the end of that tail. So the head of the sperm makes it in, nucleus can dump its contents, which is basically the genetic materials, all it really cares about getting in there. And then the flagellum that has a bunch of mitochondria in it often gets degraded and broken down. So most species, there isn't any paternal contribution towards mitochondria in the zygote. Sometimes you may see, one or two mitochondria might get in, but they're so greatly outnumbered by the tons and tons that are many copies of mitochondria that are found in the egg that we don't see it in subsequent cellular divisions as development continues along. So this is a really different inheritance pattern than with nuclear patterns of inheritance, where you can actually run your Punnett square and think about all the Mendelian stuff that we've talked about. This will run out looking really different. Okay. Let's see. So mammals are maternal. There are other organisms. There are some fungi and some yeast organisms that have some paternal or they have both inheritance patterns. And then there are plants. Yeah, Jason?
I just want to ask more of a big picture question. Why did scientists start looking at the genes associated with the mitochondria? Why is that specific?
Yeah. So what kind of diseases would you expect from mutations in mitochondrial DNA?
ATP reduction.
Big time problems, right? So if you have any issues with creating energy, we're taught from this really young age that mitochondria is the powerhouse of the cell, whatever that means, right? So then you go on and you take these other classes and you actually learn about cellular metabolism. anytime you screw with those mitochondrial genes, so if you have mutations or inherited disease patterns, yeah, so then it became an area of focus. And there are quite a few human diseases involved with mitochondrial genes, and they almost all are huge nervous and muscular tissue problems. So then people started focusing on, what's in the mitochondria. Plus, there was also a lot of interest because of the symbiotic theory of how we got mitochondria and chloroplasts. There's interest in that, comparing it. Why not study it? Yeah, there's a bunch of reasons why. But I mean, ultimately, health, disease reasons. Yeah. It became an early study to, I would say, I would add to the list of population dynamics. And this was not for humans, but for, especially in conservation, this is still widely used, which is, I'm surprised with the sequencing that's available nowadays. But folks still use mitochondrial markers to track population studies and different species that are of concern observationally. So you can use mitochondrial DNA to look at maternity and paternity, because you can show essentially who's related to who, at least through the maternal line, in fish and in birds and in other wild species. So folks have used, because it's cheap and easy to sequence mitochondrial DNA. Yeah. And it's easy to extract because there's so many copies for every cell versus one copy of your nuclear genome. So in the lab, it's a little bit easier to work with something.
How would you go about extracting mitochondrial DNA to get it?
Yeah, you would put I was gonna say, a little loud no, it's perfect. Yeah, you would focus on a tissue type that has a lot of mitochondria, like muscle. And there's kits available that extract circular versus linear DNA, and yeah, there's ways to pull out mitochondria. Yeah, we can chat later about that stuff. Yeah, there should be a lab that's attached to this, would be really good. But I think Perna's advanced eukaryotic stuff does that. Okay. The only thing I wanted you guys to know, so angiosperms versus gymnosperms. Does anybody remember plants-ish, 242 should know this? So, yeah, so gymnosperms are conifers and a fir type, you think about with pine cones and things like that, that's gymnosperms. So both of these angiosperms and gymnosperms, they've shown for both mitochondria and chloroplasts that sometimes they're paternally inherited. So there is some paternal inheritance patterns in some organisms. That's what I wanted you guys to know about. And they don't necessarily know why. Most plant sperm are not motile, or they don't have a flagellum. Okay? So it's got to be some other mechanisms of how mitochondria are being coming in from the sperm to the offspring. And that's kind of an area of research. That kind of wraps up my mitochondria inheritance stuff. Offspring are going to look just like the mother in any kind of maternally inherited pattern. All right, then. Chapter 8 stuff. Yeah.
I just want to make sure the offspring and genes together like the mom or
Okay, so maternal effect is, I hate that term because it's introduced with the mitochondrial inheritance pattern. Typically they're going to be identical still to the mother if it's a maternal inheritance pattern. And then what they're referring to phenotypically is certain genes that contribute. So it doesn't mean that, because clearly we don't all look like exact copies of our mothers. In some cases, thank God. Right, so it's talking about if you were to assay for a phenotype of a particular gene that's on the mitochondria, phenotypically that would look exactly like the mother, and genotypically, both, to answer your question, yeah. But it doesn't mean the entire phenotype of the organism, just the phenotype for that gene that's found on the mitochondria that you're interested in. And that could be like protein composition or, some other, I think your book has like a seashell, shell morphology, snail shell morphology example. There aren't a whole lot of mitochondrial genes, so they're not tons of detail. Other? Yeah.
So that's only in the general
Yeah, and that's why I don't like, it's introduced along with the concept of mitochondrial DNA in your textbook, but it's not exclusive to mitochondrial inheritance. Yeah, but it's kind of separate. That's why I almost wanted to treat it as a separate thing for you guys. Not necessarily related to mitochondria, but it is in a sense. So there can be maternal effect genes that are nuclear genes that are in a cell-by-cell type cases. Yeah. And it's unrelated to sex linked, and it's unrelated, it's not, so it's kind of a different monster in itself. And I believe you guys had to calculate something for maternal effects. I'm sure. Don't, that's not something I'll put on. Not the calculation part. Okay. Others? Sure. Okay. So, chromosomes. Well, Julie, your shirt reminded me, she's got a turkey on her shirt. And there's all these turkeys around my house, but conveniently, as November gets closer and closer, I no longer see the turkeys. So, they know. Okay, so this chapter, again, it's really short, the lecture I have for chapter 8. It's a lot of really cool stuff, but we just don't have time. Just had a faculty meeting this morning about how we should do a lab on cell biology and look at chromosomes and cytogenetics and everything, and I geeked all out on the meeting and was like, yes, let's do that. So this is a lot of really cool stuff, but we're not spending tons and tons of time on it. We did already cover karyotypes. You guys are familiar a little bit with some of the terms of thinking about chromosomes, right? So we've talked about how there can be a metacentric, when you're describing the location of the centromere, we discussed how if it's kind of in the middle of the chromosome, it's referred to as metacentric. If it's more towards one end or the other, it's acrocentric. And I believe a new term that your book comes up with in this chapter is when you have almost no little arm, the P arm at all, and it's called telocentric. Okay, so we've sort of thought a little bit about chromosomes. You guys saw some images of chromosomes. And that's what this whole chapter is kind of focusing on, chromosomal changes that can happen. So we'll start actually with more of the number piece here, how you can have variations in the numbers of chromosomes that are abnormal. And then we'll also think about structure, and in that sense, talk about whole chunks of chromosomes moving around from one to another, okay? And what this means in terms, I'll try to, as much as I can remember to point out, what this means in terms of phenotype. So do we tend to see consequences because of giant chromosomal rearrangements and abnormalities? So along with this discussion about karyotypes, another tool that cytogeneticists use is called G-banding. So you can take chromosomes and stain them, and you can actually observe this on a light microscope. You don't need fluorescent microscopy or anything like that. It's the name G-banding actually comes from this stain. It's Ginza stain. So it's actually a super easy technique. You just extract some metaphase chromosomes, you drip some of this Ginza stain on your slide, throw a color slip on, and look at it under the light microscope. And what you see, what you would actually see is literally bands, and they're of different thicknesses running along the chromosome. So I'm kind of just randomly picking sizes. And so you'll actually see light and dark areas going along this chromosome, and they make this band kind of pattern. And it's just like a barcode. So this is unique for every chromosome that you have in your cells, and same with all other organisms. Your chromosome 1 looks very different from your chromosome 2, looks very different from your chromosome 3, et cetera, et cetera. Okay? But each one of your chromosomes, number 1, your one from dad, your one from mom, look the same. Okay? And that's because, so this dark and light banding is reflecting how condensed the chromatin is. So what does that, because we talked a little bit about staining patterns in terms of heterochromatin and euchromatin, right? So what do you expect these dark bands to be? Yeah, tightly packed, wound down. You have a ton of dense DNA in that area, which relates to what? Which one, heterochromatin or U? Yeah, heterochromatin. So that the darkly stained, dense packed chromatin is the heterochromatin, and then the light areas is our euchromatin. So cytogeneticists, folks use these to identify chromosomes, for one thing, to be able to tell differences in the banding pattern. And then the other way that they can use this is to think about gene-rich areas, compare if we're going to get into times where you might have an entire chunk of the chromosome missing, or an entire chunk of your chromosome 1 has been broken and re-glued on your chromosome 15, and you can see those kind of changes based on this banding pattern. Okay, you can see that after you've done 50 years of looking at these chromosomes and know how to identify them. There's also programs and software nowadays, of course, that make computers that can make this really easy to look at. But there are folks that are experts in looking at these banding patterns, and they can just instantly tell right away. Okay, and that would, for whatever organism that they are specialized in, whether it's human or otherwise. There's actually quite a few hospitals that hire cytogeneticists, folks with my background, for example, to look at human cells for cancer cells. So we're going to talk about some abnormalities, and that's really common. Huge growth chromosomal abnormalities are really common in cancer cells. So this is a way that folks can identify and characterize chromosomes. All right, so we're going to think about some chromosomal stuff that can happen to our chromosomes. Okay? Whether it's addition of old material or removal of material or rearrangement of material, gains or losses of DNA. Yeah, Stella?
Do the dark stains always have thick bands and the light stains always have thin bands?
No, did I draw it like that? I guess I kind of did it, yeah. No, they're not, yeah, the thickness of the band is kind of arbitrary. Yeah, for that piece, you guys don't need to be the experts. I'm not going to give you a chromosome and say, tell me what chromosome it is. Go. Yeah, so that's really variable by chromosome to chromosome. Yeah, and even organism to organism. Yeah. There's a whole field of study that looks at chromosome evolution. You know, why? Because you guys we've been thinking a lot about the genome and genes and sequencing, but why are they packaged on the chromosome? Why do we have 23 chromosomes? And a chicken has 72. Or we have 46 and they have 72. I mean, so there's folks that have studied, like, why is our DNA arranged the way that it is? And why do we see these different banding patterns? Yeah. this is a chromosome, in case that's totally not clear. Okay, so this is our wild type. This is our normal chromosome. And we're gonna mess with it. Okay. So what I'm gonna do up here is sort of draw some different possibilities of what can happen to this chromosome. So you guys have an overview in your notes, sort of like an outline of where we're gonna, and then we'll talk about each one of the possibilities in more detail. The colors I'm showing are just so that we can track lengths of this chromosome if we delete it or rearrange it or things like that. But they're just representing sequences as we move along the chromosome. And I'm just breaking it into quadrants so that we can track it easily. So one of the things we'll talk about, one of the possibilities is nothing. Completely delete that chromosome, so you have a missing, the entire chromosome is missing. Another possibility that we'll go through is if you actually delete an entire large segment of the chromosome. Okay, so these are really, I don't know why I made it smaller, it's supposed to be same size. So it didn't shrink, it's just missing this black segment. So again, these are not like a one nucleotide deletion. This is like a giant megabase, on the megabase scale mutation or error. So we can be missing the chromosome completely. You can end up with a deletion. You can, another possibility is the chromosome can be here intact. Okay. Except for one segment, so in this case the blue segment, has been exchanged completely with a different chromosome. Okay, so in this case the little red piece, I'm trying to indicate using colors that this segment is from a separate chromosome, from a different, not the homolog chromosome, but a totally different chromosome. So that's actually referred to as a translocation. So you can translocate, you can break off a chunk and replace it with another chunk from another chromosome. The sequences on the chromosome can be inverted from each other, okay? So you can have, I don't know, say the black segment and the blue segment are still intact and in the correct order, but you can actually see places where, in this case, I'm flip-flopping the purple and the green segment. So instead of the purple segment being terminal, now it's an internal location. So this would be where you're cutting out double-stranded breaks, and then that piece is rotating and being inserted back in backwards, essentially. So this is referred to as an inversion. Two more possibilities. You can have a duplication of a large segment. So maybe where our green, purple, and blue segments are normal, everything's wild type about them, but the black segment is 2 times as long. Okay? So this is referred to as a duplication event, where somehow during replication, you've taken a segment of the chromosome, made a copy, and then inserted an exact copy right next to it. And our final option is when you actually take the entire chromosome and duplicate that. Okay, so then you'd have an entire, complete extra copy of the chromosome. It's like me using every color that I possibly can for this type. Okay, so this is just called an extra chromosome, and we'll talk about what that means in terms of ploidy levels. So this is stuff that can happen to your chromosomes. So again, large scale. We're not thinking about small scale changes. You have sort of this area too at the top, the deletion and the missing, where you're losing, right? So this, both of these instances are representing the loss of genetic material. Whereas the lower right-hand side, the duplication event and the extra chromosome event, you're actually gaining genetic material, right? So these are examples how we can potentially gain genetic material. And then the other two, inversion and translocation, it's more of a relocation. And we'll even talk about, so we'll get into detail a little bit more about each of these scenarios, but I wanted you guys, so this is more of a relocation of genetic material, right? So somewhere else in the genome. But I wanted you guys to have this, I don't know if these kind of things are helpful for you, but to have this kind of overview. These are the different pieces, the types of gross chromosomal abnormalities that are observed both in nature and then of course experimentally. Right? Okay. And if you're more of a visual person, then perhaps having the diagrammatic kind of stuff, you can think more about what's happening on the chromosome, what's happening in the sequences as they move around or get duplicated or deleted. Diagram makes sense. What do you think is worse? If you had to choose, if you could pick, okay, tomorrow I'm gonna have one of these things happen to me. What's worse, a gain, a loss, or a relocation? Loss, yeah, always lost. So it turns out duplication kind of events are usually tolerated better in the genome. Okay, so deletions typically are deleterious or lethal to the embryo, especially if we're talking about a huge, large-scale deletion. So depending on the size, if we're just talking about one nucleotide, but we're talking about huge chromosomal kind of differences that you can see on a microscope. So depending on how big, it can be lethal to the embryo. Whereas the big duplications can also have phenotypic consequences, but generally not so bad. So maybe in your notes you could have tolerated better, phenotypically. And then actually we see, so over evolutionary time, we see big duplications like this that are beneficial to the organism, that are retained through natural selection, okay? So evolutionarily, you see this happen naturally, and it's not only tolerated, it can be beneficial depending